Yeah, thank you very much for the introduction. So in this session, we will do the libRoadrunner interactive demo. So for today's logistics, so this will be an interactive demo session. So if you have any questions, please stop me. And due to the limited time, we will not be creating the SBML using Copasi, but we will browse and how it would look like. But if you would like to see how we are creating, and please, you can just go to this YouTube link, which is also timestamp is correct. And in this session also, we will be using the PhysiCell Studio to create the configuration file, and rest of the will be the coding in the, I will be using Notepad++ and the comment window to simulate. And also, it is going to be a slightly simple model than the OD energy model. There will be no cycling because of the time dependencies. So as an agenda, I will just touch about a little bit like the integration and the model description. I will give the model description, and then we will look for the capacity and we will code and run. So actually, so SBML is storing ordinary differential equations using the that are storing the chemical reactions between the chemicals. So in the previous session, we talk about the integration, and I will just like briefly touch this one again. So so BioFEM is doing the update and secretion, and the PhysiCell stores the internal metabolites as an amount. So then we are using these amounts as a concentration, and this cellular data will be a interface between the PhysiCell and the libRoadrunner. And the libRoadrunner is our SBML solver, ODE solver. And then we will update the SBML. And then in every cell actually has an SBML structure in it. And then we will simulate the SBML using libRoadrunner, and then we'll update the rest of the agent-based modeling. And it will be like the concentrations and phenotypic parameters. And phenotypic parameters will actually will be very important for the, of course, like agent-based modeling. And as you might remember from the yesterday's session, we are using a kind of like a phenotypic catalog. So in the XML, the configuration file, there will be a mapping between the SBML species and the phenotypic parameters or any other PhysiCell related entities. So to get just like give an example, there you can specify the death rate, and you can just map this map death rate to the apoptosis rate as using just DAA, and you can use the same structure for the other persistence time. And there are some structures that are requiring other additional information, like the index of the phase transition rate, or the which substrate we are using for the secretion or saturation. So those need to be specified in the token. And as an example, you can see that like we can change the substrate uptake rate of oxygen using SUR token. So and then we will go to the demo, but before we go that, I would like to actually introduce the model a little bit. So in here, we will have 3 different substrates in our model: oxygen, glucose, and lactate. And intracellular, there will be 3 reactions, and there will be additional species, which is called energy. And the energy will be created with 2 different reactions, the anaerobic and anaerobic, and the energy will be consumed, and there's an energy usage reaction. And then we will be using phenotypic tokens to change the migration speed, apoptosis rate, and lactate secretion rate. And the migration speed will be actually kind of like based on the how much oxygen is represented there. Apoptosis rate is based on the energy level, and the lactate secretion rate will be calculated based on the intracellular lactate concentration. And please be aware that this is a toy model, so so that the parameters are actually not scientifically estimated. It is more to represent the behavior and the integration. So the color, there will be 3 color you will see. The first color will be if the energy level is greater than 445, it will be yellow. And if the energy will be less than 440, we will see them, they will be moving and they will be motile. And if it is less than 430, then the cells will trigger their apoptosis. So usually generic workflow, we usually create the SBML first, because, or we are kind of like, we are looking for our like specific SBML, and then we are creating our model domain in PhysiCell Studio, and then we will integrate this SBML into the PhysiCell model. So at this moment, actually, we don't have any user interface for the in the PhysiCell Studio for the libRoadrunner, but it is one of our thing in our table. So let's go to the SBML exploration. So we will be using Copasi, and let's make this one a little bit smaller, so therefore we don't see so many blank spaces. So we will use the toy metabolic model. So in this model, actually, we have this compartment and the species and the reactions. I will just touch this a little bit, touch everything in it. So you might remember in the previous session, we talked about that SBML, like not PhysiCell doesn't support all types of SBML. It should be only intracellular SBML, because we are doing our transfer reactions, which are exchange rates, usually called in SBML. So we are doing this one in the bioFEM. So we don't need to do double transfer time. So the only thing we are having in here is the intracellular compartment. And as a species, actually, when we just go here, you will see that there are 4 species intracellularly, like that is important for the reactions, and which are glucose, oxygen, energy, and lactate. And there are 4, and we won't be using transition rate. There will be 3 species that will be used in the trigger event, which I will show you. And these are apoptosis rate, migration rate, and lactate secretion rate. So the reactions looks like this. So the global quantities is the where we store the constants and parameters for this SBML. And there are 3 thresholds, which are proliferation, which we won't be using, move, and death in here. And these are the constants for the reaction kinetics, which are, so if we go to here, for instance, you can see that glucose plus 6 oxygen creates 38 energy using the KAR, which is 0.01, which is just like mapped in this global quantities. And we go to the event. So this is, you might remember from the last session, this is where we are actually just changing the phenotypic behaviors or phenotypic species in here. So for instance, if we say that if the energy level is less than the energy move threshold, it will be 10 in here. So it will start moving actually, and it will create it will secrete the lactate in here. You can see that like lactation secretion rate, lactate secretion rate will be changed. So this is the SBML that is created. And again, if you would like to see more one-on-one creation, please refer to the previous ones. And let's go here. Okay, we were here. Okay, and I just want to show that, and this is how it looks like actually, if you just look in a configuration in a text editor. This is how it looks like. SBML is not like human readable, so that's why actually we are using a software to do that. So okay, and then SBML integration. So before we go to the SBML integrate in like going to just like integration, we will create the domain first. So so right now actually I have like literally a plain PhysiCell in my desktop folder. So I will first populate the OD energy sample. So the reason why I am doing, because we are using a spatial if def commands in the make file. So if you need to use the SBML integration, you need to first populate this OD energy sample and then change where you want. And so right now I will go to my desktop. So, and I will go to my PhysiCell. Okay, while I am here, I will open the PhysiCell Studio. Oh. It is, oh, okay, I wrote reverse. Okay, right now we will actually use the template first, and then actually we will copy and paste it. So right now all the energy is ready to use, but we won't be using right now. So we will be creating our domain first. So I will put this one here, maybe a little bit, make it smaller. Okay. So in the first one, actually, the first thing that is important in here, so the domain size are the same, and it is because it is a sample project. It is not like a big domain, and the maximum time is just 1440, and I will change my thread size to 10 because I can my PC can handle it. Okay, so these were the basics. So if we go to the microenvironment tab, so we will create 3 different substrate, which will be oxygen and glucose and lactate. So in here, oxygen will be having 100 micron as a cubic micron per minute as a diffusion coefficient, and decay rate will be 0 actually, and initial condition will be 38, and the Dirichlet boundary condition will be off, and the track in agents, this should be like as a default should be turned on, because this is where we are using the intracellular concentrations. How we kind of like track them. And for the glucose, it will be 300 in here, 0, 0, and this will be 50. And again, it is on and these are off. So the last one will be lactate, and lactate will be again the same diffusion coefficient with the glucose. There is no decay rate, and there is no lactate in the beginning in the model. So I just checked that. Okay. So when we go to the cell types, actually we will have only one cell type, but we will be using live cells, and it will be 0.0 as a transition rate. And when we go to the death, actually the cells, so we will not, okay, again, we will say that transition rates are 0, and there is no death. And also there will be no cycling actually. So if we go to the volume and mechanics, there is nothing to change. And for the motility, so as an initial value, we will not have any speed, and persistent time is 0.1, and the migration bias is 0.9. And the one it is important is we need to enable the motility, even though like we will, if we just simulate in this kind of like in this configuration, it won't move. But in the after we assign the migration speed, it will change. It will be changed. But in the beginning, as a default options, these are 0. The speed is 0 as a secretion. So oxygen will be uptaken in this rate, and glucose will be again uptaken. And lactate will be 0 everywhere, but there should be target value, because then we can the cells can secrete something. If we didn't put this target, then the cells wouldn't secrete any chemicals. So there will be no interactions, there will be no intracellular tab because it's not implemented yet, and there will be a custom data actually. So like we discussed in the review, so we will be using custom data as an interface between agent-based modeling and SBML. So I will just write these ones quickly. And these are the concentration, or these variable will be helping us to save the data actually. Yeah. Okay, these will be 0 as in the beginning, but we will change in our coding part. Okay, if we go to the user parameters, I will actually kind of like change this one as an initial internal oxygen. So this should be double and 0.8. Again, initial internal glucose. So this is kind of like user parameters are for the like a parameters for the PhysiCell. It's not just bound with the cells. So we will be using these values as a reference value while we are seeding the cells, and they won't be used after we use actually. Okay, so user parameters are done. Okay, let's check and save it, but let's save it. Okay, so I will save. Sorry, I will save as actually, and I will be kind of like overwriting the PhysiCell settings what we have, and yes please, and I need to open the yes, and I think I don't need to do that because we will be going to the coding part, but let's see. Okay, if we check this one, everything looks okay, except what I didn't, I forget this actually. The saving output will be in every 30 minutes instead of 60, so we will see more fine results. Everything looks good, but the one important thing that we are missing is we don't have any intracellular attribute yet. So what we will do is we will add this attribute into the I will explain this one into the phenotype. So it can be anywhere. Of course, you don't have to like split the attributes. So I'm just putting in phenotype attributes. So these will be here, and I will explain these ones in a minute. Okay, so what we are seeing is here is the interface, and we are using intracellular configuration, and the type is libRoadrunner, and we are specifying the SBML file name, and it is in the actually this is the what we have looked in the Copasi, and there's an intracellular parameter which will be just, if you watch the previous session, this is how often we evaluate the intracellular models, and these are the kind of like a mapping between the PhysiCell substrate and SBML species, and there is a PC phenotype species which will be using our tokens, and this is the death apoptosis rate, and the apoptosis rate is a species in the SBML that is changed by the events. So in here, what we will doing is kind of like we will simulate our like agent-based modeling, and then we will change the SBML. SBML will be updating, and if the energy level is less than some threshold, then this apoptosis rate will be changed, and then the cells will start to die. So we can specify any kind of like those listed phenotypic parameters, and in here actually also we will be changing lactase excretion rate as well as the migration speed. Okay, so we will remove the intracellular related parts in this custom modules in a minute, and then we will build one by one. So therefore we can see that, okay, what is going on in here. And so let's go and do that. So this is the custom CPP, but this is in the sample project, so this is not the one we like. So I will go to the custom modules, and this is the custom modules we are changing. So in here, there are actually two parts that is important for the intracellular integration. The first part is in the setup tissue. While in setuping, we need to start the intracellular, and also we need to do the housekeeping for the important values. And also there is a one update intracellular function, and we will be kind of like removing inside. So these part will be actually kind of like important for us. But I will explain this one in a minute. And this is the big update intracellular. So what you will see is, maybe I can just briefly look here. So this is a void function that is just turning in every cell. You can see that this is paralleled, and it is doing the intracellular workflow for every cell. So, and it is parallelized. So this is a good thing. So we will be just removing here. So, okay. So until here, if you came here, or if you miss anything, the ground zero is our zip actually kind of like will be just have exactly the same setup that I have, that you can find in the codes. Okay, so this was kind of like was the starting. Okay, and then so we have the model domain. We created the model domain, and then so we know how many cells in it actually. So it's kind of like, if you just go to the setup tissue, we will see that there is a kind of like a circle tumor cells, which will be based on the initial tumor radius, and then it is a kind of like a cell spacing, and according to the cell radius, it is creating a circle in the middle of the simulation. And then we are kind of like assigning the position based on the calculated position with the cell circle positions. And then we need to now we need to seed, while we are seeding the cells, we need to add the setup tissue related intracellular related setup tissues things. So first thing first, let's do the first part. So what we are doing in here, okay, we actually we seeded the cell. So this is every cell. Actually, we are just seeding one by one, and then we are setting the single behavior of custom intracellular oxygen based on the initial internal oxygen user parameters, like I explained in the while we are creating. And in the so therefore we kind of like one by one we change the single behavior of custom data according to the initial one, because this is the first thing we need to do. And in the second part, we will actually we will again doing the similar thing. So actually this is a kind of like a redundant line. So I'm literally doing the same thing in here. But what we are doing is, okay, we could change the custom data, but still PhysiCell molecular internalize are not kind of like changed. So therefore we are just getting the signal of custom data we assigned in here, and we are multiplying by the concentrations to change the total substrate, the total amount of specific substrate. So this is a important thing in PhysiCell. In PhysiCell, if we track the substrate, so this is how we are kind of like reaching them, and then we change the intracellular, sorry, molecular amounts. Okay, after we seeded, and then we need to start the intracellular. And in here, because cells are already kind of like have their own specific SBML, this is the creating SBML structure in every cell, and we are just changing the energy level according to the intracellular energy level, which is the initial one. So this was actually kind of like a housekeeping and the first thing that we need to do. But still, so imagine that like in our main CPP, so we are kind of like, okay, we created the domain, we created the microenvironment, we created the cells, and this is how we are creating, how we are setting the tissue. And then after that, actually, maybe I can just show, it might be much better. So so this were kind of like, we set up the macro environment, we create the cell types, and we set up the tissue. And then we are just going kind of like saving the regular, the first initial SVGs and we are entering the main loop. And in this main loop, like we are saving in here, and then we are making our diffusion, and we are doing the update all cells. And this is the one thing that is special to the SBML and intracellular also. So we are updating our intracellular function in here, which will be kind of like this function. Okay, so now we are coming to the full integration. So the first thing first, in this part, the microenvironment will be using, sorry, the bioFEM is updating the intracellular amounts, which is actually automatically done, so we don't need to do this one. But in the second part, we will be kind of like calculating the custom data based on how much internal metabolites amount we have. So this is the first part. So, and also for the later usage, we needed to use the cell volume for the later. But so this is kind of like, again, housekeeping. But what we have done is we are getting the intracellular concentration just using get single signal, and we are changing we are having oxygen value. So this is a double. And we are having the glucose value based on the intracellular glucose and lactate. And so we kind of like did this part actually. So we did the second step, And if we just go to, and then, okay, we have the values, and then let's update the SBML, and let's update the SBML. So what we are doing is set parameter value is a special function for, actually I have kind of like discussed this one from the previous session, but this is a special function that helps us to update the SBML value. And in here, the oxygen species in SBML will be set to the oxygen value intracellular in here. So we are just changing the SBML one by one. And then we will run the SBML, we will update, we will do the ODE simulation. An SBML simulation looks like this. Are all cells in this indices? So this is the pointer. So this cell is updating their intracellular, which is just like a one iteration of ODE solving. And then there is another specific function for the SBML, which is called update phenotype function, which is just looking for the tokens that we set, and then it changes the parameter, phenotypic parameter based on the given mapped item. Okay, so we have done the update, the run evaluation of the OD model. And then we will go back in here and we will update the intracellular values based on the SBML. So this looks, I know, kind of like heavy, but we will go one by one. So in here, internalize chemical update. So as we discussed, actually, we kind of like we are changing this this part, internal metabolites amount, because imagine that like we have in our OD, there's a lactate that is created by the intracellular reactions. So therefore we need to change the intracellular concentration, and we are changing accordingly. So we are getting get parameter value is a function that helps us to get the parameter, get the value from the SBML, and we are multiplying by the cell volume. And then this yields us at the total amount, and we are changing the total amount in here. And as a saving purposes, we are using custom data, and we are again updating these values based on the get parameter, oxygen, glucose, lactate, and energy. Yeah, I think this should be it actually. So this was the what we put together, and we can simulate it. So before we do that, I need to compile. Okay. Okay. So in here we have 144 cells that are in our simulation, and we can just look actually in the output folder. This is the initial seeding looks like. So this will take a little bit time. So while I'm doing it, actually I will show a one kind of thing that because like right now we can't so the PhysiCell Studio still the intracellular kinetic OD is still not perfectly integrated into the PhysiCell Studio. For the post-processing, for the post-analysis, we will use a different a kind of like code that we actually Randy wrote. So I will go to desktop and PhysiCell. So I am in the PhysiCell folder in here. So we have a plot cells.py. So there's a like Python function that we can just visualize the cells, and also there is a data analysis for this one too. So what it does actually, it is kind of like a preliminary version of in the PhysiCell Studio, and it is still in 6 hours. So we will wait a little bit more. So we don't expect any like division, so because we set the value. So what I just want to look in here. Okay, and so I will so this is the kind of like the final model, and if you just download the final model.zip, you will actually reach this point. Let's see where we are. We are nearly kind of 40%. Still, cells have lots of energy. They don't have, like, they didn't consume their energy, so they are just like happy cells in the happy microenvironment. Okay, we reached at some point in the middle of the domain. The oxygen and the glucose are actually kind of like consumed that much, so therefore the cells doesn't create their energy that much, so therefore they kind of like fall down in the energy threshold. So then they need to, we will see that they will start migrating, or they are trying to escape from that area. Okay, yeah, you can see that. And then there are some unlucky cells that are in the middle that are actually already started to dying. So I will show in a substrate level too, but I just want to show in the cellular level first to make it more clear. Okay, so maybe I can just use the data right now. Okay, in here, this is a kind of like overlapped version of the cells and the substrate. So in here, what we will see is oxygen is getting consumed in the middle of the domain. And then what you will see, it's the same for the cells, and the cells are trying to escape from the middle, and some of them are dying because of the lack of oxygen. So maybe we can just look for the glucose too. And it is kind of like a similar behavior for the glucose. Again, the glucose is getting used in the middle of the domain. And then cells will start do the same behavior. But let's actually look for the lactate. You might remember in the beginning, we set the lactate secretion rate to 0. So if we don't specify any secretion rate, so they shouldn't secrete any lactate. But in the SBML, we are changing these values. So let's see. Okay, so cells are secreting their lactate because the SBML is kind of like changed the lactate secretion rate, and then they started to secrete their lactate. Yeah, it took around 5 minutes, but also I was doing so many stuff, so it should take around 4 minutes usually. But yeah, this is a kind of like a full integration. So what we will do next, so yeah, this is what we are seeing. What we will do next, okay, what if we change the DT intracellular? So okay, I can just close this one. Okay, to do that, actually, we will just come here and then we will change it. So what we are doing in here, instead of simulating intracellular model in every diffusion DT, we will be simulating in every phenotype DT. So let's make data cleanup. And I don't need to do make, but let's be safe. And then let's simulate OD energy. So actually the thing is, do we see the one important question is, do we see the differences or not? So let's, while kind of talk while we are having our simulation, imagine that like we have a pool that is just like kind of like updated in every 1 minute. That is just like we are uptaking some chemicals and then we are using intracellularly in every 1 minute. So what 0.01 minute. And then right now what we are doing is we are just like simulating our diffusion every, again, 0.01 minute, but because we are dealing with the reaction rates, so even though we change the intracellular, so the only thing it will be just like the removing part or utilizing part will be just like big bunch of similar usage actually, and this will be kind of like a big 6-minute simulation, and then it will just go down. So allegedly, of course, like this should actually kind of like increase the simulation speed, because we are not doing the OD simulation regularly. So this is more less simulation for the intracellular one. So the question is again, will we see the difference? And if we see, so this means that our model is kind of like is not converging in this DT. But if we don't see that, if any differences, then this means that okay, our model is not that complicated, so therefore it is not kind of like getting affected about this DT intracellular increasing. And I will use plot data for that one. Or maybe, yeah, let's see. Okay. Again, oxygen is getting consumed. We can see that. And let's go to the lactate. Also, lactate is getting excreted. Oh, we are actually seeing a same effect. So it started around, as far as I remember, like 14 hour or like 13 or something. We are seeing the same behavior. So so this means that actually our network is not that complicated. So therefore it can be simulated in every 6 minutes without any kind of like convergence. But again, this is a toy model. So therefore it is kind of like understandable that like it doesn't have any complex behavior in it. So yeah, this was actually kind of all. So what we have done in here, instead of kind of like giving the parameters, or instead of giving a static phenotypic parameter in the PhysiCell, the phenotypic parameters are controlled by the molecular OD model. So therefore it is a dynamic change, and we didn't write so many stuff. We just like put the integration, and then the SBML is keeping this dynamic changes based on the intracellular amounts. Yeah, this was actually kind of the last thing that I want to show. So thank you very much for our funding agencies, NIH, and this was a kind of like developed by the in the project of NIH, which is in this ID. So this was kind of like integrating the molecular models to the PhysiCell to understand the colorectal cancer and CAF metabolic relationship. And also I would like to thank my lab mates and Dr. Finney's lab and Dr. Momenter's lab and other collaborators. And yeah, thank you very much. And if you have questions, actually, we have 10 minutes for the questions. Yeah, thank you very much. If you don't have any questions, actually, I think I can stop the recording.